Good morning, everyone, and thank you for being here today. I am pleased to present to you a detailed overview on the qualifications involved in liquid chromatography mass spectrometry, or LCMS. Today's session is titled Qualification of Liquid Chromatography Mass Spectrometry, LCMS, and it's designed to give you a clear understanding of this powerful analytical technique. Over the course of this presentation, we will explore the fundamentals of LCMS, its applications, and the key qualification processes to ensure accurate and reliable results. Let's get started. Today, I want to introduce you to liquid chromatography mass spectrometry, or LCMS for short. This technique is a powerful combination of two methods that work together to analyze complex mixtures. First, liquid chromatography separates the components of a mixture based on their interactions with the stationary and mobile phases. Think of it as a way to sort out the different molecules so we can study them individually. Then, mass spectrometry comes into play by analyzing the separated molecules, providing detailed information about their mass and structure. What makes LCMS so valuable is its ability to identify and quantify compounds with high sensitivity and specificity. This makes it a fundamental tool in many fields, from pharmaceuticals to environmental analysis. Overall, LCMS embodies the core theme of our presentation, precision and analytical power, by combining separation and detection in a seamless, efficient way. Let's take a closer look at liquid chromatography mass spectrometry, or LCMS for short. This technique combines two powerful analytical methods, liquid chromatography for separation and mass spectrometry for detection, into a single, highly effective system. Typically, an LCMS setup includes several key components. First, there's the LC pump, which moves the liquid sample through the system at a controlled flow rate. Then, the autosampler automatically introduces the sample into the system, ensuring precision and reproducibility. The column oven helps maintain consistent temperature conditions to optimize separation efficiency. Next, we have the ion source, which can be electrospray ionization or atmospheric pressure chemical ionization. These are methods used to convert the liquid sample into ions. Finally, the mass analyzer, such as a triple quadrupole or a QTOF, measures the mass-to-charge ratio of these ions. For example, a typical mass spectrometer might cover a range from 20 to 2000 mZ, and it's designed to keep the mass drift very minimal, less than 0.1 to over a 24-hour period, ensuring reliable, consistent results. This combination of liquid chromatography and mass spectrometry allows us to separate complex mixtures and detect compounds with high sensitivity and specificity. It's widely used in fields like drug quantification in plasma, impurity profiling, metabolite identification, and even pesticide or food contaminant analysis. Overall, LCMS is a versatile, powerful tool that plays a crucial role in modern analytical science, enabling us to analyze complex samples with precision and confidence. AIQ, or Analytical Instrument Qualification, is a critical process that ensures we have reliable analytical data. When we talk about data reliability, we're really emphasizing that the results we get are accurate and consistent, which is essential for making informed decisions. AIQ also helps us maintain regulatory compliance, meaning we stay within the rules and standards set by authorities. This is vital for passing audits and ensuring our products meet quality requirements. Another key aspect is inspection readiness. By thoroughly qualifying our instruments, we're always prepared for inspections, reducing the risk of delays or non-compliance issues. Most importantly, AIQ demonstrates that our instruments are fit for their intended use. This means they perform as needed for specific tasks, whether it's testing purity, potency, or stability. Now, referring to the USB 1058 model, AIQ is broken down into several key stages. Design qualification, or DQ, which confirms the instrument is properly designed for our needs. Installation qualification, IQ, ensuring the instrument is installed correctly. Operational qualification, OQ, verifying it operates properly under specified conditions. And finally, performance qualification, PQ, which proves it consistently performs as required over time. Together, these stages create a comprehensive approach that supports quality, compliance, and confidence in our analytical processes. This framework is integral to our overall goal of maintaining high standards and ensuring our instruments are always ready for use. Let's take a closer look at an example URS, or User Requirement Specification, to see how it fits into the design qualification process. The main objective here is to verify that the instrument's design actually meets its intended use, ensuring it performs as expected. In the design qualification, or DQ, we focus on activities like vendor selection, which is critical because choosing the right vendor impacts the quality and reliability of the instrument. We also review the URS itself, making sure it clearly captures the user needs and requirements. Additionally, we verify the technical specifications to confirm they align with the URS and support the intended use. Highlighting these points is important because each step ensures that the instrument will perform reliably and meet regulatory and user expectations. Connecting these activities to the overall validation process helps us build confidence that the design will truly meet the specified requirements and ultimately support successful validation. Let's walk through an example checklist for verifying the correct installation of an instrument. The main goal here is to ensure everything has been set up properly and is ready for operation. First, we focus on the installation qualification, or IQ, which is a crucial step in confirming that the installation meets all the necessary requirements. Checks under IQ include several key areas. The first is hardware installation, where we verify that all components are correctly assembled and securely in place. Next, we look at electrical supply verification, ensuring that the power connections are appropriate and stable to prevent any issues during operation. Environmental conditions are also critical. They need to be within specified ranges to ensure accurate and reliable instrument performance. Then, we move on to software installation, confirming that the software is correctly installed, updated, and functioning as intended. Finally, proper documentation is essential, making sure all records of the installation process are complete and accurate, which supports traceability and future audits. Overall, these checks form a comprehensive approach to validating that the instrument is installed correctly, aligning with our broader goal of quality assurance in every step of the process. Let's take a closer look at the main goal here, which is to verify that the instrument performs as intended. This is a crucial step to ensure reliability and accuracy in our results. When we talk about acceptance criteria, one key aspect is the operational qualification, or OQ. This involves a series of tests designed to confirm that the instrument operates correctly under specified conditions. Some of the specific tests we perform include checking mass accuracy, which ensures measurements are precise and within acceptable limits. Resolution tests confirm that the instrument can distinguish between different signals effectively. Sensitivity assessments help us understand how well the instrument detects low levels of analytes. System precision checks verify the consistency of results over multiple runs. And calibration verification is essential to confirm that the instrument remains properly calibrated over time. All these tests together form a comprehensive validation of the instrument's performance, giving us confidence that it will deliver reliable data in our ongoing work. This step is fundamental in maintaining quality and compliance throughout the entire analytical process. Let's talk about performance qualification, or PQ for short. The main goal here is to verify that our analytical system performs reliably during routine analysis. This isn't just a one-time check. It's an ongoing confirmation that everything is working as it should. Key components of PQ include system suitability testing, which helps us ensure the system is working correctly before analyzing samples. Then, we have calibration curve verification to confirm that our calibration remains accurate over time. We also analyze QC samples regularly to track the consistency and accuracy of our results. Reproducibility checks are crucial too. They help us see that our results are consistent across different runs and days. Now, regarding how often we perform these checks, it varies. Some are done daily, especially before starting routine work. Others are done per batch to ensure each set of analysis maintains quality. And of course, periodic verifications are important to catch any long-term drifts or issues. Connecting this to our overall quality approach, PQ is fundamental because it ensures our method remains valid and trustworthy over time, supporting the integrity of our data and compliance standards. Let's take a closer look at some of the typical LCMS system suitability parameters. These are essential to ensure that our system is performing reliably before we start any critical analysis. First, we have retention time reproducibility, which is all about making sure that our analytes consistently come off the column at the same time each run. This consistency is crucial for accurate identification and quantification. Next is peak area precision. This refers to how consistent the measured peak areas are across multiple injections, ensuring our results are precise and reproducible. Then there's the signal-to-noise ratio, a key indicator of sensitivity. A high ratio means our system can detect analytes even at low concentrations without being masked by background noise. Finally, mass accuracy is vital for correct analyte identification. It ensures that the masses we measure are close to the true masses, giving us confidence in our data. Together, these parameters help us verify that our LCMS system is suitable for analytical work, providing reliable and accurate results that support the overall integrity of our study. Now, let's take a closer look at this calibration curve example. As you can see, this type of curve is essential for quantitative analysis because it allows us to determine the concentration of unknown samples based on their measured response. Here, we've plotted some example data points, which illustrate the relationship between known concentrations and their corresponding signals. The key idea is that once we establish this curve, we can use it to accurately find the concentration of any sample by simply measuring its response and interpolating along the curve. This process is fundamental in analytical chemistry because it transforms raw measurement data into meaningful, quantifiable information, tying directly into the overall theme of precision and reliability in our analysis methods. Let's now talk about the limit of detection, or LOD, calculation. This is a crucial step because it tells us the smallest amount of analyte that we can reliably detect with our method. Essentially, it helps us understand the sensitivity of our analytical technique. To determine the LOD, we typically look at the signal-to-noise ratio, often using a ratio of 3:1 as a standard. That means our signal from the smallest detectable amount should be 3 times higher than the background noise. We also use statistical methods, such as calculating the standard deviation of blank measurements, to make this process more precise. Connecting this back to our overall goal, knowing the LOD ensures that our method is capable of detecting even trace levels of our target compound, which is vital for accurate analysis and compliance. So, understanding how we calculate and interpret the LOD helps us validate the robustness and reliability of our analytical process. Now, let's talk about the limit of quantification, or LOQ. This is a key concept in analytical methods because it defines the lowest amount of a substance that we can reliably measure with acceptable precision and accuracy. Essentially, it's the point at which our method can produce trustworthy, reproducible results. Understanding the LOQ is crucial because it helps us determine the sensitivity of our analytical technique. If the concentration of the analyte is below this limit, our measurements might not be dependable, which could impact the validity of our data. In the context of our overall presentation, the LOQ ties into ensuring the quality and reliability of our results. It's about knowing the boundaries of what our methods can accurately detect and quantify, so we can interpret data correctly and make informed decisions. Remember, establishing a proper LOQ is fundamental for compliance, especially in regulatory environments, where accurate quantification can be critical. So, always consider the LOQ when designing or evaluating analytical procedures. Let's talk about precision. When we focus on precision, we're really emphasizing the importance of accuracy and exactness in our work. It's about making sure our measurements, data, and actions are as close to the true value as possible. This is crucial because small errors can lead to big differences down the line, especially in complex projects. Precision helps us build trust in our results and ensures consistency. Throughout this presentation, we've seen how precision ties everything together. It's the backbone of quality and reliability. So, mastering precision is essential for achieving the high standards we aim for in our work. Now, let's talk about the documentation requirements that are essential for maintaining compliance and ensuring smooth regulatory inspections. First, we have the User Requirements Specification, or URS, which clearly outlines what the system needs to do from the user's perspective. This document is the foundation for everything that follows. Next, we have IQ, OQ, and PQ protocols. These are crucial validation documents. Installation qualification verifies that the system was installed correctly. Operational qualification confirms it operates as intended, and performance qualification ensures it consistently produces the desired results. Calibration certificates are also vital. They provide proof that instruments and equipment are properly calibrated, which directly impacts data accuracy. Maintenance logs are equally important because they track all service activities, helping us demonstrate that the system remains in good working order. An audit trail is another key element. It records all changes and activities within the system, providing transparency and accountability. Lastly, system suitability reports give us evidence that the system is performing correctly before we start any critical analysis or production activities. All these documents work together to ensure compliance with good manufacturing practices, or GMP, and they are essential during regulatory inspections. Proper documentation not only helps us maintain quality, but also builds confidence with regulators that we are operating in a controlled and compliant manner. So, in conclusion, LCMS qualification is essential for ensuring reliable analytical performance, which means your results are consistent and trustworthy. It also helps you stay compliant with regulatory standards, giving you confidence that your processes meet necessary guidelines. And of course, it guarantees accurate quantitative results, which are crucial for making informed decisions. The key steps in this qualification process follow a logical progression: DQ, IQ, OQ, and PQ. Each stage builds on the previous one to solidify the system's performance and compliance. Regular maintenance, calibration, and thorough documentation are also vital. They help you maintain regulatory compliance over time and ensure your system continues to perform at its best. Overall, this structured approach and ongoing effort are what enable labs to deliver precise, reliable, and compliant results consistently. For further support, please don't hesitate to reach out to us. We're here to help clarify any questions or provide additional information you might need. You can contact us through the details provided. And finally, I want to sincerely thank you for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, we're always available to support your next steps.